Okay, if you guys are okay, we'll get going. As I mentioned to you earlier, today will be a slightly longer than usual class. I have some things to maybe think about for your upcoming term test, and I'm hoping that you've all been spending some time studying. I'll come back to this slide again toward the end, but just to, again, remind you that your upcoming term test, which is next week, you won't be in this room. You will all be in EX320. You will actually have sort of a 1-hour test, and then I think it'll take you about 20 minutes to do that sort of again in groups. You can decide like what groups you want to be in. You don't have to decide that now. On the day of the test, if you want, you can also form groups on the spot. It is worthwhile studying for it. You won't be able to use any other resources. Your only resource will be each other, and collectively you'll come up with the answers that you have just seen in terms of the questions. So we'll do that next week. I'm hoping it's a good experience for all of you as well, and it will be during class time. We'll probably be getting started at about 10:15 next week, just to give you a few extra minutes to get in there. Today's lecture is on the term test, just to let you know, and actually there's a fairly large amount of coverage, both in terms of the short answer questions and the one-line questions that you get on term test 1. You might think it's only 3 lectures, it won't be so bad. If you haven't been studying, I'll talk about that at the very end. Again, you don't need to know any of these different titles on this slide that you are seeing here, but it's just to give you an indication that it's a really important discovery. CRISPR-Cas9 is one of those technologies that right now is a very dominant technology that we want you to have some idea in terms of how it works. We know that there are a lot of things that are being developed related to CRISPR-Cas9, and in fact, one of the reasons why I picked this group of papers is to show you that this individual here at the bottom, Eric Olson, in this science paper that wasn't published too long ago, actually sent up in advance the paper that I'm going to be talking to you about a little bit later on. So there is a lot of strategy in terms of publications and other things to be aware of as well. So we've been looking at this dystrophin complex, and we've been really focused on dystrophin itself as the molecule. When you don't have dystrophin, which acts as a shock absorber that kind of links the sarcoplasmic membrane, or the sarcolemma, with the actin-myosin cross bridges within different muscle tissue. And you have to keep this in mind because you're going to need to know this for next week as well. When we talk about actin-myosin, and we talk about what actin and myosin does, we've been concentrating on skeletal muscle, but can you tell me what other muscle types also have actin and myosin fibers? So cardiac muscle has actin and myosin, and we need to consider that as well. Any other muscle types that you know that have actin and myosin, going back to PSL 301 or 300? Does smooth muscle actually contain actin and myosin? Actually, it does contain actin and myosin. It's just not in a regular repeated array. And so when we talk about this later, dystrophin is not just a complex that's associated with one type of muscle. It is actually involved in many, many different tissue types. And when you look at a paper, you need to understand why they're looking at different things. So one of the reasons why we're looking at dystrophin as an important sort of regulating molecule that's found between actin and myosin and the other extracellular matrix molecules, and it acts as a link. We know that there are many, many mutations that occur that result in a premature stop codon. Some of those are very, very bad for individuals that have it, and we're thinking if we could somehow bypass that premature stop codon, which is very common in genetics, maybe there might be a way to go back and help individuals that kind of have this disorder. I have talked about this, and I'm going to come back to this diagram. This is color-coded at the top, is sort of the regions that we look at for the protein. So both in mouse and human protein, the regions that are encoded by the genes down here in the same color, so for example, the actin binding domain regions, are also highlighted in yellow as these different genetic exons that you are seeing here. When we have these different exons that are lined up between 2 all the way through to 8, you will see that some of these actually are sites where we have exon acceptors or splice sites that are splice acceptors, and we also have these splice donors as well, the different shapes that you see here. So keeping in mind that we can't just skip any specific sequence, they actually have to form a functional protein. When we have like exons and we're trying to delete some of these exons, it doesn't always work the way that we think. If we were to look at these blue regions, for example, between 36 and 37, these are just simply exons that aren't alternatively spliced, and so we can easily eliminate exon 37, and we can join 36 to 38 because there are no splice sites, there are no exon acceptors or splice acceptors and splice donors that we would see as we did earlier in this sequence or in the sequences that we have over here. So this is sort of where we're at right now. We have this big protein that's incredibly important for muscle function. Some individuals have mutations that cause a premature stop codon to be generated, leading in a truncated or smaller non-functional version that can't appropriately link between actin as well as dystrophin. All of that is what you need to know for next week. It will all be recapitulated for you on the term test as well. So here we are back in the MDX mouse. And again, this is another theme. Do you know all of the models that we have looked at throughout this sort of 3-lecture series? The MDX and the MDX52 mouse models are incredibly important, and Eric Olson's group has been using this MDX model system quite a lot. One of the reasons why they are interested in the MDX model, and again, is it a good model? How many of you, based on what you've been studying so far, because I know that's all you've been doing every single weekend, Saturday, Sunday, you've been looking at HMB360, studying in advance. When you look at this, does the MDX mouse look like it has muscular dystrophy? It does not. It does not have all the different features in muscular dystrophy. They have a normal lifespan, but they do have a premature stop codon in exon 23. And again, this is where we need to understand that not all exons can be simply cut and paste back together. In this particular case, if we're looking at exon 22, it actually doesn't have any splice donor or acceptor sites on the side of the exon that immediately comes after it. And so if we were to somehow eliminate exon 23 that has a premature stop codon, the next exon in line, this one here, 24, can simply reanneal and then form a functional subunit. Will you be missing one of the domains? You will, but it'll be still a functional protein because of this. And this is sort of Eric Olson's idea to be able to use this mouse model to set up for a future study that he just published to be able to do some CRISPR-Cas9 mediated exon skipping. So if we bypass or skip over this exon that has this premature stop codon, will we get a functional form of dystrophin, and will this MDX mouse be able to express dystrophin to make it look more like a normal mouse? One thing to keep in mind, this system here, we actually have to cut this exon twice. So we're cutting in this intronic sequence just before exon 23, and we are also cutting in the intronic region over here. Keeping in mind that we have two different guide RNAs to cut two different regions of DNA. Now, again, if you have been following what I've been telling you, what is one of the advantages to allow that we want in terms of genomic editing? Why is this better than TALEN, for example? This is a good example of what sort of phenomenon, one that you need to know for the upcoming term test. Yes.
You don't have to design two TALEN molecules to directly attack both sites, so you don't need 4 in total to attack.
So I don't need to design 4 separate things. That's definitely one reason why we don't need to worry too much about this. There's one other reason. Yes, at the back.
TALEN can only snap one DNA strand at a time.
Right, so TALENs definitely only target one DNA sequence at a time. We need a duplex, or we need a dimer of the FOC1 enzyme for sure. Anything else? Yes?
Multiplexing?
So here's a good example of multiplexing. By simply having different guide RNAs and using the same Cas9 nuclease, as long as we have a guide RNA for this region, and as long as we have a guide RNA for this region, we can use the same Cas9 enzyme to cut both strands of, or both regions of the DNA. And in fact, this is the way in which Eric Olson has set this up. There are different guide RNAs, a specific guide RNA, and then a different guide RNA that sort of is used to cut at a separate site. This is why we have two. One for the exon itself, and then one sort of outside of the exon. So if you're wondering why there are two guide RNAs, we're making two cuts to completely eliminate. This entire exon will be gone. Now keep in mind, this is only 2 years ago. This is the way in which technology rapidly advances. And I want you to keep this in mind for this type of strategy that you see here. It is very likely that this year, unlike last year's term test, you will get a picture similar to what you see here, where you'll get different exons, and then you will have to tell me, based on this study, as well as the study that comes after, which approach you would actually use in your short answer. And again, just a little bit of a preamble to that. So on the bottom, we know that in order for us to deliver this to muscle, we need to use viruses, and we use an adeno-associated virus. These are myotropic. And one of the reasons why we use these myotropic viruses, we needed to specifically target muscle cells themselves. So again, just keeping all of that in mind, and I'll talk a little bit more about this SP-Cas9 in a moment, and why this is important. We then went last week, before we finished a bit early, looking at the different ways in which we could actually inject these two different constructs. One adeno-associated virus, which contains the nuclease, and then we have another adeno-associated virus that contains the two guide RNAs. One guide RNA for the MDX premature stop codon, and the other one that is just outside of the exonic region to make those two separate cuts. They decided to look at intraperitoneal injections, intramuscular injections, which makes the most sense, and then this retroorbital behind the eye injection that we saw this movie on at the end of the last week. And again, they follow it for different lengths of time to make sure that the expression of dystrophin is changed and is maintained after this injection. So we went through this slide very quickly, and I wanted to go back over this. So we are looking at the staining of dystrophin. This is what we would normally see in a wild-type mouse. This is not an MDX mouse. This has normal levels of dystrophin. This is what we should expect to see. And even though there is no physiological or pathological change that we can observe, so its muscles don't get any smaller, weaker, etc., it has a normal lifespan, unlike what we see in muscular dystrophy. We see a lot of that red staining disappearing in the MDX mouse model. If we go back in and we use CRISPR-Cas9 to eliminate that premature stop codon, so it doesn't form dystrophin, and we can see that dystrophin is almost completely gone. There is no red staining. That if we go into this MDX mouse, age matched, same age, born at the same time, but now being treated by delivery of the CRISPR-Cas9 using intramuscular delivery, and we look at the same overall muscle tissue, which is part of the forearm, the tibialis anterior muscle, then we can see that a lot of the expression comes back. It's not quite at the same level as the wild type, but it is much, much better than what we would see under normal MDX conditions. I'll also tell you that one of the things that you should know is, how do we look to see if there is some pathology associated with it? We take the same tissue that we've done this staining for dystrophin on at the top in red, and we can look at what is known as hematoxylin and eosin, and you can simply use the short form, which is H&E staining. It is a histopathological stain that you can do on muscle or any tissue. And we will often see on H&E staining necrotic tissue. So when we see tissue dying through necrosis, we can identify it fairly easily in H&E. The other reason why we use H&E stain is to also examine whether or not we get these tiny purple or dark purple cells. So normally the background is pink, as you see here, and it's a little hard to see on this diagram here, but we can use H&E to look for what are known as inflammatory cells. We can't identify what type they are, but we can see more of these inflammatory cells in the dying tissue. So it's a good way to see if there's inflammation in a tissue. It's a good way to see if there's an inflamed and group of inflammatory and immune cells that are entering into damaged tissue in a quick sort of analysis. We don't have to stain for specific cell types. So again, because there is a GFP tag on the virus itself, on the guide RNA and Cas9 molecule itself, we need to see that all of these effects are actually occurring because we have genomically re-engineered this tissue. The other reason why we want to be able to use this is if GFP is being expressed, and again, apologies on this, I know students don't really like it when we do this, but if we're looking at GFP, this green fluorescent protein, one of the things that we need to know is, without doing any staining, if it is being expressed, how long is this going to be expressed for? 2 weeks after we've injected into the muscle, we can see there is a lot of green fluorescence. Do we see it going down, for example, after 6 weeks? So another month goes by, and in terms of a mouse, that's a few years worth of its life. And when we're looking at the overall expression, we actually see that it is maintained or at slightly higher levels with time. And we see that when we look at dystrophin, that we also see a better sort of response for dystrophin. So this shows a proof of principle that using CRISPR-Cas9, not only do we have maintained expression of the guide RNAs and the Cas9 nuclease, but in fact it actually helps in the repair process where we actually see a better recovery of the dystrophin molecule here on the bottom. And again, it's pretty remarkable, or at the time it was really remarkable to be able to say that all of those different issues that people had identified, well, you know, it's a really interesting technology, but you probably will have to keep going in and injecting every few weeks to maintain a certain level. They had shown that once infected, it stays infected for a long period of time, and in fact, the recovery improves with time. You don't have to go back and actually re-inject that tissue. Did you have any questions about anything that I've covered so far, sort of as a recap, or anything that you are a little confused about, or anything that isn't clear for you at this point? There's going to be a lot of this in next week's term test. A lot of things that I've just talked about, things that you're going to have to apply. So if you have any questions, or if you aren't sure about them, maybe circle it now, come and see me during the break. Sorry, there are questions. Yes?
What's used to stain for dystrophin?
We use an antibody against dystrophin, the protein. So we have to go back in, provide the primary antibody and a red secondary antibody to stain for it. Luke, you had a question.
Yes, would we be able to inject the Cas9 and the guide RNAs in the same virus, or would we need two separate?
So that's a great question. So I had mentioned in passing, because this is a slightly older paper, even though they do the same thing in his most recent paper, nowadays we would want to have everything in one virus. We have two separate viruses, one with the Cas9 at the top, and then another one with the guide RNAs in a separate virus. We inject them into the same tissue, hoping they're going to target the same tissue. Based on the results that they have, it certainly looks like it works because we see GFP, that's on one construct, and then we see the dystrophin coming back. And in order for that to have worked, we would need both of them coming in and infecting the same tissue. Nowadays, we would want to have that in the same construct, going into the same virus, but at the time, this is what they wanted to do. But you're correct. In many ways, we'd actually want them to do this experiment with within one virus with guide RNAs and the Cas9 all packaged into the same construct. Any other questions on this before we go on? Yes?
Why did the two viruses in the previous slide as well, they have different
Promoter systems?
Yeah.
Okay, so I'm going to come back and talk about this, but in order to get the best expression, because we're using two separate types of viruses and we're making use of the transcriptional and translational machinery, one, we need a general promoter that you're seeing here. I'm not showing you all of the data. I will on the next paper that I'm going to cover later. But because of the translational machinery, we actually don't want to have the same promoter system overwhelming it, because these are going to be expressed at much higher levels to begin with. So compared to the normal proteins, after you get an infection, this will kind of take over most of the cellular machinery. If you have two competing for the same sites, whether it's the U6 or using the CMV or the cytomegalovirus promoter system, you're going to actually overwhelm the translational machinery, so you may not get efficient expression of both. It sort of goes beyond the scope of what you need to know, but I thought it would be good to answer that. Any other questions that I can answer for you? Yes?
Was there any significant difference depending on the type of delivery, IP versus IM?
That's a great question. Which one is better? So I'm going to come back and address that in a few slides. Great question, by the way. So in terms of what we're seeing, we are seeing better expression. They actually did a really cool thing in this particular paper. These details you don't really have to worry too much about. This goes back to the actual way in which the gene in MDX mice is set up. And one of the things that you'll see is we have a premature stop codon, and we are going to cut this exon completely out using CRISPR-Cas9. So imagine like you have a piece of string, and that piece of string has some damage to it. So you're going to cut around the edges and reseal that piece of string back up, which is sort of outlined for you here. And again, if you look at the PCR fragment, because you have eliminated this exon, the PCR that you generate is going to be much smaller, which is what you see here. We know that it is equally loaded because when we use the GAPDH as a control for our PCR, that we have pretty much equal loading of the transcript. So we're using this real-time PCR, and we are generating this secondary transcript here at the bottom. And you might think, oh, okay, proof in principle, I see that. I see the original genomic sequence, and then when I take out the muscle, which is what is done here, and then I reverse transcribe and I amplify, I also see that there is some repair that is being done. This is all taken 4 weeks after the muscle has been injected with CRISPR-Cas9. It's not completely edited, so we still see some of the original genome up here, and then we see some of the corrected genome down here at the bottom. But one of the really cool things that they did was when they saw these smaller bands down here at the bottom, where they think that exon 23 is being eliminated by cutting around it, they actually took the gel, they cut the gel out, they cut this PCR fragment out, and they actually sequenced that PCR fragment, which is what you are seeing over here. So not only are they confirming on a gel that it is smaller, kind of what you'd expect, you eliminate one of the exons, of course the resulting PCR fragment is going to be smaller as you'd expect, but they actually took the time and effort to go in and take one of these different PCR products down here at the bottom, and then they actually sequenced it, which is what you're seeing here on the right, and there is a clean sort of re-annealing of exon 22 with exon 24. It's like exon 23 never existed, and in fact it was an incredibly cool result to be able to see that through through sequencing the PCR construct, that they were able to actually show that it was indeed fully corrected. Exon 23 is completely gone, and just as they hypothesized, exon 22 was able to re-anneal or reattach with exon 24. It was a seamless experiment, and it was, again, really brilliantly done. This had sort of reattached itself or respliced itself with exon 24. Again, these were long-lasting changes that were observed. And again, when you take a look at the different tissue types, I'm showing you tissue from skeletal muscle. They were also looking at cardiomyocytes or cardiac tissue as well. In this particular case, they are seeing that with time here on the y-axis, that with the MDX, as the mouse gets older, there is more and more necrosis. This is the necrotic autofluorescence. So these dystrophin molecules are degenerating, the cells are degenerating, and eventually over time there's nothing left, which is why we see less of this after 12 weeks. But after we inject, and this is retroorbitally behind the eye, we're actually seeing that we get an improvement over time. So similar to the results that I had shown you before. So again, this technology seemed really, really amazing. The fact that we could genomically engineer things using CRISPR-Cas9. The sequencing results show that it worked exactly as the theory had predicted, and functionally they were actually seeing an improvement. Now here is one other thing to think about as well. So when we look at these mice, one of the things that we don't typically talk about is that we see a loss in muscle tissue. We don't see that they live any less. They live about the same time as wild-type mice. But if we're looking at, for example, more dystrophin, which means that the skeletal muscle is more intact and it's able to contract the way that it should, and there's fewer and fewer of these necrotic degeneration events, then one of the things that we should be able to see is a change in muscle function. So again, even though they live about the same length of time as the wild type, so this is the wild type, this is the amount of force their muscles can generate. So if you take a little weight and you apply it to their forearm muscle, which is what we've been examining, generally speaking, let's say they have about 75 grams of force that they can actually apply to a weight. The MDX mice, even though they don't have any overt behavioral changes, when you actually measure their muscle function goes down dramatically, and it is much lower. So let's say about 35 grams of force. If we were to inject in and we saw that there's less cell death, there's more dystrophin, does it result in a functional recovery, which is something we always want to see, not just biochemical, not just genetic evidence, not just histological evidence, all of the previous slides, but do we also see a behavioral change in terms of muscle function? We also see that there is a change. It is not at the same level as wild-type mice, so there is a statistical difference between wild-type and following CRISPR-Cas9 mediated editing, but it is significantly better than what we would see in just MDX alone. Whether you're looking at male mice or female mice, so you might expect that there's a hormonal element to all of this in terms of muscle mass and muscle function. It doesn't matter what mouse you're looking at, they are equally improved after delivery of this CRISPR-Cas9 system. Again, any questions on this before we go on? So behaviorally, if you were asked, you just have to look at some index of muscle function, like how much weight it can support, or how much tension it can generate, or the number of grams of force that a muscle will be able to generate. So one of your colleagues were asking this a little bit earlier. So in terms of all of the different routes, so whether it's intramuscular, whether it's retroorbital, or injected into the stomach, all of these were able to improve the muscle function in different organs as well as muscle groups that were far away from the injection site. So you inject in the stomach, the muscles in the arms improve. You inject behind the eye, the muscles in the arms and legs improve. Or if you inject directly in the muscle groups will improve. And again, the dystrophin expression in these MDX mice were improved in a number of different tissues, including in vascular smooth muscle cells. It's also improved in smooth muscle tissue, and this occurred through all routes of delivery. Interestingly, the most effective route was actually through retroorbital injection. I had asked you this last week, so here's how good your class memory is. If any one of you can answer this, I will be most impressed. It is not listed as one of the different traditional routes here, but if I were to inject behind the eye, retroorbitally, and this is the best way to actually see expression of smooth muscle cells being enhanced, what is that analogous to in the human situation?
Easiest to find vasculature.
Yes, but if you're looking at the route of injection, what would we call that in humans? Is it subcutaneous? Is it intraperitoneal? Obviously not. What do we call that? What is it similar to?
Intravenous.
Do you agree with your colleague it's an intravenous method of delivery? Yes, no, maybe? So if you agree with your colleague at the front, you can do a hard blink now. Not a lot of blinking. Okay, at least you agree with yourself. That's good. Your colleague is right. This is an intravenous route of delivery. There are a lot of blood vessels behind the eye, and there are a lot of venous blood vessels behind the eye. So when you get those bloodshot eyes after studying all night Sunday for your term test on Monday, and you come to your term test, I'll walk up beside you and say, "Thank you for studying hard," and then also, "Hey, I like your veins." So again, just to keep that in mind, it is intravenous. There's a lot of blood supply behind your eye. And it seems to be the most effective, as your colleague was asking, especially for targeting vascular smooth muscle cells or smooth muscle cells in general. Interestingly, because of the way that AAV, remember, we're taking CRISPR-Cas9 in the guide RNAs, we're packaging them into a virus. The virus infects the cells within the tissue, the muscle cells within the muscle, for example, but it's targeted specifically toward the muscle. And I'll get to that in a moment. It cannot cross the blood-brain barrier, so this is unlike some of the other adeno-associated viruses that we'll see later. And in fact, we also see dystrophin. So if you were asked on a multiple choice test question, do we see dystrophin in neurons? Yes, we see dystrophin in neurons. So we see it within the hippocampus, in the CA1 region of the hippocampus. But we don't actually get improvement of dystrophin levels within the hippocampus. It is excluded from the brain. It only targets muscle tissue, whether it's smooth, cardiac, or skeletal muscle. They get targeted. The brain is still at a deficit for this, and in fact, we don't really know the true role of what dystrophin is doing in the brain. I had mentioned at the very outset that for a variety of reasons that we still don't understand, even though it targets muscle, that in fact there is a cognitive decline. These individuals don't do well. They're not quite as at the same level of brain development as individuals that have the full dystrophin gene. We don't know what it is doing within the neurons. That's something that maybe some of you might want to actually explore. But we do see it, but the viruses, they can't cross. Therefore, there is no improvement within the brain regions that we would expect to be able to see. So again, in terms of what all of this means, there are a couple of really important implications. This means that using an adeno-associated virus, AAV9, that targets muscle tissue, we can actually do this. We can repair a damaged gene. We can go back, we can show this on multiple levels, biochemically, genetically, as well as behaviorally show that we can go in and fix this. This is also long-lasting. It was a proof of concept that, in fact, we don't have to keep going back in, which is what everyone thought, that after you inject it once, you'll have to go back every single month and inject this into a person because the gene expression will disappear. Cas9 will go away, the guide RNAs will go away, but they showed a long-lasting and very persistent expression of all of the gene editing components. By using this cutting method, by excising an exon, and because it was able to repair itself through the two mechanisms that you should be very comfortable with for next week, where if you simply cut, the DNA repairs itself. We cut it on one side and we cut the exon on the other side. Those two repair itself by non-homologous end joining or NHEG. This is actually much better, or NHEJ, sorry, much better than simply providing a template that will actually go in and fix that stop codon that was prematurely mutated. So this is actually a proof of principle, at least in their hands, that rather than creating an oligonucleotide that could give the correct sequence, and by doing what is known as homology-directed repair to fix that gene or that stop mutation, instead of trying to fix it, this is a better method in terms of the efficiency in which the muscle tissue gets expressed. Interestingly, at the end of this particular article, Olson mentions this. He didn't actually show this directly, but he showed that if you could edit muscle tissue using NHEJ methods, so simply causing CRISPR to make a cut in DNA, double-stranded cut, that you don't actually have to do anything by providing a template. So this is the non-specific modification, that any kind of insertion or deletion, so anytime you cut DNA at a region, especially if we're looking at a donor or acceptor sequence, which is not what he did. Remember, he's not looking at a donor, acceptor, or acceptor sequence. He's just looking at a normal exon that, in fact, you can generate exon skipping. This sets up his second paper, which is what he published again in Science in the summer, or just as we were starting to return back to school, related to this particular topic. So again, this is Eric Wilson, and this is now following up on his original paper. This is basically the last bullet point statement that you see here. He didn't actually do this in his first paper. He hypothesized that this could be possible. He didn't do it, but he was setting up his second paper, which he likely had in the works anyways in his lab. He was probably already doing it, and he was able to do that experiment here. So again, we'll just go through a few slides and then we'll take a little bit of a break. Much of this is sort of a review for you, so you should kind of know most of this. So I'm hoping that, again, it's not anything brand new for everyone in this class. We know that there are a lot of different mutations that occur within the dystrophin gene, specifically in Duchenne's muscular dystrophy. And this can, again, cause degeneration, necrotic loss of cells, and eventually the atrophy or loss of this muscle tissue. And this can affect more than just skeletal muscle, because we know that it can also affect cardiac muscle tissue as well. It can also affect the respiratory skeletal muscles that help you to breathe, and this is sort of the two major reasons why individuals with Duchenne's muscular dystrophy, it is a lethal mutation in them, and they often don't make it past their teenage years. We've already looked at the structure. I haven't asked you to memorize it, nor do I expect you to know this number of 79 exons, but if you go back to all of the slides that we've had so far, you'll see that the very last exon is number 79, and we see that in both mice as well as in humans, and we'll see the same thing again within dogs. In that earlier paper that I just showed you, without referring back to your notes, so let's see how good your very short-term memory is, so roughly on 30 minutes, without going back, which exon were we looking at in Olson's MDX paper? Exon 23. So great, you guys have great short-term memory. I hope that you all have great long-term memory. We'll find out on Monday. So that's a great model. We know that MDX by itself is not perfect, but if you take a look at what happens in the human condition, we often see that these mutations aren't occurring within exon 23. It happens spontaneously in that MDX mouse model. But if you were to look at the human condition, you would actually see that many of these premature stop codons are found either in exon 45, 46, 50, etc. And this means that when you're looking at all of these, that ultimately it leads to the loss of the normal sort of reading frame by the time you reach exon 51. And this leads to a shortened or out of frame protein, which means that it's not functional, it doesn't work, and that's the normal human condition. So even though we have a great mouse model, those mouse models are sometimes very limiting, and we've already talked about why there are some limitations. So if there's some way to do exactly what was done by Eric Olson's group earlier, and we can somehow bypass things getting gummed up or stopped at exon 51, that would more closely resemble what happens in the human condition rather than just concentrating on exon 23. So again, when we look at Duchenne's muscular dystrophy, there is a naturally occurring spontaneous mutation. Unfortunately, so I have a dog, and I know that my dog, when I did some research on the Shih Tzu, that also has had, through inbreeding, a lot of these different genetic mutations. And in fact, because dogs have undergone so much breeding over a short period of time, it's no surprise that we have a lot of these different mutations. And one of these spaniels had sort of a naturally occurring mutation that occurred after breeding had taken place, and that was transposed into a beagle model, which we now kind of use. So I talked about Cocker Spaniels earlier on and dog models, and this beagle model is sort of the one that we're going to be looking at in this canine system. So this canine beagle model system that has the Duchenne's muscular dystrophy mutation, it has a lot of the same clinical features and pathology that the human disease has. So it has that muscle weakness. It's not where you have to actually apply a weight. So if you look at a mouse, you won't actually see any change in the way that it walks around or anything else. You wouldn't behaviorally be able to distinguish them until I actually apply weight to the DMX mouse or MDX mouse. You wouldn't actually be able to see it. But in these beagle models, you can actually visibly see signs that their muscles are degenerating. They have atrophy or loss of muscle tissue, as well as fibrosis. So you lose muscle cells. We get fibroblasts moving in. So we have inflammatory cells moving in, we have fibroblasts moving in, and eventually all the muscle tissue will be replaced by fibroblasts, which means they are not contractile. They will form scar tissue, as we saw a little bit earlier on. So this canine model system, where we have a change in exon 50, will lead to many of the clinical conditions that we see within the human condition, and this is why it becomes important. So this model system kind of looks at what is going on here. A lot of the same ideas that we looked at earlier. So this is exon 51. You can see right away that this is a splice acceptor site. So there is that odd shape that you see here. So this would be, for example, a splice donor site. And this is a splice acceptor. It would not be able to seamlessly cause a reading frame that would allow 49 and 51 to be accepted with each other. And the idea is, if Olsen is correct, we don't need to actually provide a template. We don't need to use a fixed piece of DNA to get past this mutation. If he was right from his previous science paper, using the naturally occurring or non-homologous end joining mechanisms, if we are looking at a splice acceptor or a splice donor site, which is what we are looking at here in this diagram, if we can somehow cause an insertion or a deletion, if we snip the DNA using CRISPR-Cas9, we should actually be able to induce exon skipping. And this was sort of his theory. And this is sort of the outcome of this particular paper. So here is that single site that is indeed a splice acceptor site. If his theory is right and we can somehow snip this with CRISPR-Cas9, we would be able to see endogenous repair mechanisms that allow for exon skipping to occur, because this will cause the exon to simply be bypassed. So again, if you're taking a look, one of the things that seems to be important, this is sort of the way in which this is sort of set up. In the in the beagle model, exon 50 is gone, so exon 50 is no longer there. And in exon 51, there is this other issue that occurs. And if we were somehow able to target exon 51, so exon 50 being gone in the beagle model, which is why we don't see it here, this can't adjoin with 51. 49 and 51 exons cannot join to each other. If we could somehow cause exon 51 to have an insertion or a deletion event that will endogenously fix it, maybe it will allow for the splice site to be repaired by itself. And this is exactly what he was looking at. So he targeted what is known as an exonic splice site, and this is why we have this specific sort of structure to this. By causing an insertion or a deletion, just simply cutting it with CRISPR-Cas9 on a region here, this actually causes it to go back in frame, and it allows it to actually make a full-length protein. And I think I'm just going to stop here for the time being, so we'll take a bit of a break. We'll come back in about 10 minutes. We'll continue on. I figured it out, so I don't mind it as much.
It's under courses. Yeah. And then click all courses. Yeah, and then you can choose. No, you can just choose to not have it on your dashboard. You can't really do anything. Because it says massive.
Okay. If you don't mind, we're just going to go on a couple of really key differences here, and I want you to be aware of this. This beagle model is missing exon 50. It is naturally occurring. Exon 50 is gone from this beagle. This is the reason why it doesn't actually form something downstream. Exon 51, again, as was mentioned on the previous slide, being one of the important issues. Here, we are not looking to eliminate exon 51. The last paper, what happened was we used 2 different guide RNAs to selectively target the regions outside of exon 23, and we used the Cas9 nuclease to remove that sequence, and so exon 23 was completely eliminated. Here it's a little bit different. We're only using 1 guide RNA, and this was sort of his hypothesis. We could still do myo-editing, and we can do myo-editing because Cas9, the nuclease, when it cuts double-stranded DNA, if it does it appropriately, it can actually modify a splice acceptor / donor site. This is a acceptor donor site, and it modifies it so that it will actually no longer have that exonic splice enhancer region, and so it looks like any other exon, which allows it to kind of re-anneal or reattach to the preceding exon that comes before it. And this is sort of his rationale. It was a proof of concept. He could do this because in the beagle model, the structure that you see here actually had occurred. Just a couple of things to, again, keep in mind. This is 1 guide RNA that is targeting this specific exon splice region. When you go back into what we looked at last week, yeah, not to make it too scary or too difficult, you are going to have to know a little bit more in terms of the detail than last year's term test, especially when you're looking at when the nuclease itself. A nuclease always has, for a double-stranded break, we need 2 enzyme cutting sites, whether we have 2 FOC1s or we have a RUBC versus an HNH region in the nuclease. You need to also know like what strand will contain like the PAM sequence, why the PAM sequence is important. And I left this one up here just as a reminder that some of your questions actually involve knowing what this protospacer adjacent motif is and whether this is important or not, and whether it's going to be targeted by RUBC or the HNH domain. Just because of the specificity of some of the questions, I don't want it to be a surprise for you on term test 1, so I just wanted to remind myself, to remind you to go back and study those specifics. Just as we saw earlier, when we look at the structure of dystrophin within the muscle tissue, and we're looking at, is this screen shaking or is it just my eyes? Maybe it's just my eyes. So when we look at the structure and we look at dystrophin, instead of being stained with a red antibody, this dystrophin is now being stained with a green antibody. As we saw previously, when you don't have this, without any type of treatment, you would have like almost none of the dystrophin around. if you were to, for example, inject this, and this was actually a really cool thing about this paper. I inject the muscle on one side, so I injected ipsilaterally. So the same side that I actually did the injection intramuscularly of the virus, similar to the last paper, I will actually see pretty good recovery, which is what you see here on the right. However, if you look at the opposite side muscle, so say I injected the muscle on the right, and that muscle on the right is what we're seeing here in this diagram. The last paper actually showed that the results were like it didn't matter where you looked. The results were very generalized. In this model system, for the dog anyway, for this beagle model, if you look at the opposite muscle actually doesn't improve. So remember the last experiment, I can inject behind the eye, I can inject in the muscle, I can inject in the stomach. The muscles generally improve. Here it's specific to the muscle that I'm actually injecting. So if I inject on the right, and this muscle would normally atrophy and die, it gets rescued, but the one on the left does not. So it's a very different and very specific effect that we are observing. So it's different. The study is a little different, and so is sort of the resulting findings that here, the intramuscular injection does not seem to be generalized. It seems to be very targeted to the different muscles that we're looking at. So again, just keeping that in mind as well for the upcoming term test. Know the differences between the muscle, the major findings in the muscle after CRISPR-Cas9 in the MDX mouse model versus the beagle model that we just gone through. So again, I'm going to take you through sort of the last part that I need you to be aware of as well. There is a little bit more neurobiology, but I also wanted to introduce some different concepts for you as we go along. And I'm just going to give you an overview of like why Fang Zhang's study is interesting, although this year we're probably not going to concentrate on it. I'd rather switch gears, go back to autism, the mouse model in autism, how CRISPR-Cas9 reverses the symptoms in autism, even though they're fully developed and even though they're fully there in the adult, how can you reverse some of those different symptoms? So again, we've been looking at CRISPR-Cas9, and these other slides you don't need to worry about in any way or form. I'll tell you what you need to concentrate on, mainly because there isn't a lot of time before the upcoming term test. Toronto actually is one of the hotbeds. I don't know if you are aware of this or not. There are a lot of researchers who are working with CRISPR-Cas9, different ways to modify CRISPR-Cas9 to make it a better system. And Toronto researchers actually had a way to turn it on and off using specific types of proteins. The other thing that I wanted you to be aware of, nowadays there's a lot of introspection about different types of papers related to genome modifying techniques. This paper was actually a really interesting paper that came out. It's not that old, so it's about a year old. There was this rush because everyone wanted to get involved in CRISPR. CRISPR was the hot thing. And this paper had actually come out saying that there's a simpler, faster, easier way to use CRISPR. And because of the nature in which people want to publish, because this is a very hot technology, because there are lots of patents and therefore a lot of money that's associated with it, a lot of biotech firms simply jumped on, you have a new paper out, we're going to invest a couple of $1 million in your research just to make sure that we are the first to be able to benefit from your research. And this biotechnology firm that was on that previous headline had actually backed this particular paper that had come out from Chen Yuhan's group in China. And it was one of those really interesting papers. There's this other DNA-guided method that would be useful. It kind of involved proteins that might normally be associated with RNAi, the Argonaut system. And he developed this faster, cheaper, more efficient way to actually do genome editing. And so people were like, wow, this might be the next version of CRISPR. And so this is sort of the research institute where he did all of his research out of, and people were really incredibly excited by this. People were jumping on CRISPR bandwagons. Everyone was going out and kind of investing a lot of time and effort into doing this, including Feng Zhang, by the way, including Jennifer Doudna and Emmanuel Charpentier. Everyone was kind of in this big race. The problem is this paper got retracted within 6 months because no one could actually reproduce the results, even though it was published in a really high-ranking journal. Part of this is because the science is so incredibly competitive. It is a race to be the first one to publish, the first one to get the patent, the first one to get the bragging rights to all of these different papers. And so when we look at how this applies in neurogenomics, so this is where we go back to what our course is about in terms of neurogenomics. I had mentioned earlier that within neuroscience, at the time that CRISPR was coming out, there were very few papers that actually dealt with the CNS. There's a lot of issues with this because when you look at the brain, we've already seen this, some viruses that you inject peripherally just can't make it up into the brain. There's a structural barrier, the blood-brain barrier, that prevents the brain from being targeted. And that's really what we want to look at within the context of neurogenomics. In addition to that, when we look at the brain, there are two competing neurotransmitter systems. One is excitatory and one is inhibitory. In fact, the vast majority, about 80%-ish, so there's a lot of debate about this as well, most of the excitatory neurotransmission that occurs, or neurotransmission that occurs, is believed to be excitatory. And again, most of this is through a neurotransmitter that is released called glutamate, and this seems to be the dominant hypothesis. By the way, what is the other 20% inhibition? And it's through GABAergic processes. Which one is more important, by the way? GABA? Inhibitory is more important than excitatory? Agree, disagree? Any ideas? Or is it simply just one of those trick questions? It is indeed one of those trick questions. We actually need both, and they're both important at different points in time. Later on, when we talk about temporal lobe epilepsy, you'll understand why I said it the way that I just did. We need both excitatory and inhibitory, and these are really important. So we know that glutamate can work either on G protein coupled receptors, where a neuron will respond very slowly, it won't be excitable right away, and we also have glutamate working on ionotropic receptors, which make the responsiveness of the neurons immediate or very, very rapid. So we have these two different forms of excitatory neurotransmission. One very, very slow because it works through a metabolic pathway, has to activate a G protein. That G protein then has to accumulate cyclic AMP or do something else like activate protein kinase A, and then it will eventually cause some kind of ion channel to be modulated, and that's very, very slow versus very fast mechanisms that involve ionotropic receptors. And these are the ones, for the most part, that people are studying and have been studying for the longest period of time. So again, we have these two different responses. This is a much faster response. This is milliseconds, even though it looks like it's the same time scale. This is in minutes. And so when glutamate is released, if we have ionotropic receptors, we get this immediate, fast, millisecond response. And if glutamate is released and it is working on metabotropic G protein coupled receptors, we get this much slower response that takes like minutes for us to see and lasts for a longer period of time as well. It's a lot of background for you. I don't need you to worry too much about that, nor do I need you to worry too much about this particular diagram here. We can measure how fast or how slow this is through a process known as whole cell recording. You won't be asked a single question on this slide, I guarantee it. It's just to let you know how we actually do this. And it also tells us about what types of receptors we have. Is it an ionotropic receptor because we see it fast? Or is it a metabotropic receptor because we see it really slowly? And so when we look at these ionotropic receptors, through genomic cloning, by the way, we are able to identify that those fast millisecond responses occur because the cell will have receptors that can bind to glutamate really quickly. And these fast ionotropic receptors are known as AMPA receptors, or NMDA receptors, or kainate receptors. And all of these bind glutamate. When glutamate binds to AMPA, there's a fast response. When it binds to NMDA, there's a fast response. When it binds to kainate, there's a fast response. And they're all named different things because genomically, we have different genes that encode for these glutamate receptors. So the genes that make up AMPA receptors are different than the genes that make up NMDA receptors, and they are different than the ones that make up kainate receptors. And they're also named after the compound that will activate them. So glutamate binds to all three of these, but AMPA will only bind to AMPA receptors, NMDA only binds to NMDA receptors, and kainate will only bind to kainate receptors. They all bind glutamate. When they bind, there's a really rapid, immediate response that we look at. And for the most part, people have been looking at AMPA as well as NMDA because those two receptors are thought to be important for learning, memory, for you to be able to have different drug responses, for you to modulate whether or not you're going to be addicted to something quickly or not. Those two receptors that are glutamate receptors are the ones that are believed to be important in this learning, memory, addiction, synaptic plasticity mediated response that you might have heard about in some of your other courses. So this is a really busy slide, and I don't need you to memorize a single thing on this slide. It's to just give you an idea of why, in terms of the neurobiology, we have an extra sort of complication. When we look at a muscle cell, so dystrophin, for example, within a muscle cell, it is part of a complex. And this complex, from dystrophin to dystroglycans to actin and myosin, all of these things are important. But when we look at the structure of the synapse, which is part of a neuron, it is even more complex. It's about an order of magnitude more complex, and it's also much more dense. So if we were to look under an electron microscope, it is so dense that it actually forms an electron density, which we call a postsynaptic density. It is really hard to introduce genes into this network of proteins that you see here. And I just wanted you to be aware of this limitation that we have and why we need to consider these limitations. And this postsynaptic density that you see here is one of those reasons which make it incredibly challenging. Again, nothing on this slide that I want you to take away. It's simply to give you a little bit of context for why, in terms of understanding genomics or understanding things within the nervous system, it's not that neuroscientists are not quite as bright as developmental biologists or anything else. It's just that this complex, this array, makes it really hard to study it, and teasing all of those elements apart has been a very, very big challenge. So this is sort of the reason why we have been a little bit more delayed compared to some of the other sciences in terms of our understanding, being able to manipulate the synapse, etc. So again, on this particular slide, I only need you to know a few things, and I don't need you to memorize everything on this slide, nor do I need you to know the structure function on this slide. There are a couple of key points that I want to give to you. These are re-emphasized on a few bullet points. So if we were to take a look at the NMDA and AMPA, which are glutamate receptors, overall what we would see is that they have this kind of structure that you see here in the middle on this particular diagram. And this diagram shows us that the AMPA and NMDA can both bind to glutamate, and this glutamate binding, which you see here in orange, is an important and key effect that we will see. Even though they both bind glutamate, so both AMPA binds glutamate and NMDA receptors bind glutamate, there is a couple of key differences that I want you to make note of. One is that when glutamate binds, NMDA receptors, after the binding of glutamate, will allow the influx, which means from the outside to the inside of the cell, or the inside of the neuron, the passage of calcium ions. And it seems to be unique to NMDA receptors. So when glutamate binds, to example, or with AMPA receptors, we really don't see the influx of calcium in the same way. So NMDA receptors are permeable or allow calcium to enter into cells after glutamate binding. AMPA receptors do not. Do you have any questions about that? I'm hoping that some of that is a review for you. There's one other aspect that I want you to jot down and to make note of as well. I don't need you to memorize everything that's down here on the bottom left, but I do need you to be aware of like two things. One, both AMPA and NMDA receptors have 4 subunits. They always have 4 subunits. We don't see 4 subunits here, but you can sort of get the idea that maybe 4 of these things come together in this type of structure that when we look at this diagram is sort of a cutaway of what we would expect. So AMPA and NMDA both bind glutamate. AMPA, when it binds glutamate, does not allow calcium, it only allows sodium to flow through, and NMDA will allow calcium to flow through when glutamate binds to it. Both AMPA and NMDA have 4 different subunits, and these subunits make them unique and have different properties that we can measure through that technique that I mentioned to you earlier, which is known as electrophysiology. You do not need to memorize this diagram, nor do you need to know any specific details outside of that. Do you have any questions on this before we go on, or anything that you want to make sure or double check on before we move on to the next few slides? Yes?
Does NMDA only take in calcium, allow calcium in, or also sodium flow?
It also allows sodium to flow through as well. Any other questions? Or anything that you want to just double check on before we move on? Yes?
Do we need to know about the magnesium blocking?
No, you do not.
These receptors?
Nope. No, you do not. Okay. So this goes all the way back to 2014. So 2014 is where we haven't really had this huge influx of CRISPR papers or Cas9 mediated papers. So I've also told you that neuroscience is actually a little bit delayed in terms of how it reacts to different types of things, mainly because there are a lot of technical considerations that make study of the nervous system, like the postsynaptic density, much more challenging than it would be in other organ systems. Again, not to say that the skeletal muscle system is easier to study, but there are some significant challenges that are unique to the nervous system and the brain in particular that make it challenging. So when you look at these papers, why is there a 2 to 3 year delay in terms of getting something out related to CRISPR? This was the first paper that came out related to CRISPR. And this was kind of a unique paper in a variety of ways. All this paper is showing is that we can do this in the nervous system. That's it. This paper is published in a very high profile journal. This is the same type of journal that we would have in Cell. Our version of it is called Neuron. It is one of those papers that is very highly cited. It's one of the best papers in neuroscience. And all this paper is really saying is we can do this in the nervous system. Is that a cool result? I guess so. One of the other reasons why this got published is because the senior author, Roger Nickel, is actually one of the most important people in neuroscience before he stepped down as the director of neurobiology at UCSF. So again, Roger Nickel's group had been studying the hippocampus and the glutamatergic system within the hippocampus and these postsynaptic density proteins, including AMPA and NMDA receptors, for probably 20 years. He did not want to get scooped. He saw this technology coming out. He wanted to be one of the first to publish in it, and so he devoted most of his assets to trying to get the first publication out on this. One of the things that he does, which is very different, so unfortunately here at U of T there are very few labs that can actually do this. Instead of growing dissociated cultures, which is what my lab used to do and many of the labs used to do here, or still do here at U of T, he actually takes a slice of brain tissue, he keeps all of the connections in that slice, and he grows the tissue as a slice. This is called an organotypic slice culture, and he is an expert in growing the hippocampus as a slice culture. So it keeps all of the structure of the hippocampus intact. One of the things that he is also really well known for is the fact that he can deliver genes to this structure. So he's been studying the hippocampus, AMPA, NMDA receptors. the brain itself has been really challenging to study, and he uses a method which is known as biolistics. Because he is an expert in recording from these different neurons within the brain on these slices, he is able to see if he can modulate a change in either AMPA or NMDA. And that's really all he is doing in this particular paper, using classic neuroscience theory and this technology that is just being developed. So Fong Zhang's paper in 2012, 2013, he's waiting and seeing sort of the results. He wants to be the first to be able to publish on this. So this slide, if you're not in neuroscience, is a good one to know. There's some elements of this on your upcoming term test. So term test 1 will have different elements of this. I told you that AMPA and NMDA receptors always have 4 subunits. And that's absolutely true. Both of them bind to glutamate, also true. We can measure that through electrophysiology. NMDA receptors that have 4 subunits always have GluN1, and we need 2 of those. We always have to have GluN1. It's a prerequisite to an NMDA receptor. And then the 2 additional subunits, we can have GluN2A or GluN2B. So we would have, for example, GluN1, GluN1, and then GluN2A, GluN2A. That makes a full NMDA receptor. So his idea was a really simple one. Do you really need the GluN1 subunit in a neuron for the NMDA receptor to function? Classic neuroscience had told us that you need to have 4 subunits and you need to have GluN1. If you don't have it, the receptor won't function. So he wants to use CRISPR-Cas9 to eliminate GluN1, the gene, and therefore the protein, and then see, does the cell still have NMDA receptors? So that's his first question. The second question is kind of related to this. AMPA receptors, the other type of glutamate receptors, are also composed of 4 subunits. They are a little bit different. So you can have GluA1, which is the gene that makes up one of the subunits, or GluA2, another gene, separate, distinct, that makes up the AMPA receptor, GluA3, GluA4. You always have to have 4 of these. But generally speaking, most AMPA receptors will contain GluA2. One of the things that I just told you earlier was that if you look at an AMPA versus an NMDA receptor, when it binds glutamate, NMDA allows which ion to flow through? Calcium. Does AMPA allow calcium to flow through? It does not. And the reason why it does not is because AMPA receptors have 2 of these GluA2 subunits. The GluA2 subunit undergoes what is known as RQ editing, and so it does not allow calcium to go through. Since most AMPA receptors have the GluA2 subunit, it means most AMPA receptors don't allow calcium to flow through. And his question is, if I were to somehow remove GluA2, which normally blocks calcium from entering the cell, can I actually see an AMPA receptor now start to allow calcium to flow through if I remove this subunit that normally prevents calcium entry? Really simple questions, nothing really earth-shattering. People had sort of looked at this and studied it in different ways, but because he wanted to be the first to publish and he wanted to have a good journal citation on this, he actually examined it using these 2 simple questions that I'm going to take you through. Do you have any questions on this before we go on? Or anything related to like the AMPA NMDA system before we go on? Any questions? So I can guarantee you next week, based on this slide here, you will get at least 2 questions. It's a good one to go back and review. And again, think a little bit about what we are discussing here. So Roger Nichols' group actually uses a really interesting technique which is known as biolistics. So we have been talking about making viruses that contain a construct, and these viruses will attach to the cell surface of whatever cell we want to infect, and it will infect those cells. Another way to do this is through what is known as biolistics. So we take biological tissue and we shoot bullets at them, and these bullets are in the form of these small gold nanoparticles. And this is Again, a fairly old technique. It was one of the first nanotechnology techniques that were developed. We still use this. It's not as expensive as you would think. These gold particles aren't going to make you rich. So if you're thinking, I'm going to join a lab that uses gold nanoparticles and take them home with me, you would have to stay in that lab for about 10 years before you have enough to make a couple of $1,000s. So just saying that these gold nanoparticles are not worth your time and effort to join a lab for if that's what you're thinking. So one of the things that makes this really unique is that we can take these gold nanoparticles, which are inert substances, and we can actually get them to attach DNA. So we can, for example, attach different types of DNA on these gold nanoparticles, combine them in a mixture, we put them into what is called a gene gun, and we put it over the tissue that we want, and we simply shoot the tissue, we pull a trigger, literally, and it will use a gas injection system, and it will deliver these bullets to the nuclei of the cells, and it will target the DNA directly into the nuclei of the cells. And as a result of this, that cell with that nuclei having that DNA, that will allow the DNA or the gene or whatever we're delivering to it, in this case CRISPR, the guide RNAs as well as the Cas9, it will actually enter into the cell and it will affect the cell just like viral delivery would do. It is a very effective technique if you know how to do this well. And again, Roger Nichols' group is actually an expert in using biolistics to change gene expression within the hippocampus. Any questions on this before we go on? So you should be comfortable with how biolistics works, what are the components of biolistics, and how you might use biolistics, not only in the hippocampus, but maybe also in muscle tissue. Again, just a little bit of things for you to think about. So again, without going into this in a lot of detail, and I'm not going to spend a lot of time on the next slide either, but when we look at the experiments, so he does this using CRISPR-Cas9. He uses guide RNAs to specifically cause DNA strand breaks in the GLUN1 gene. And when he does this, is he able to demonstrate that his tissue no longer expresses NMDA receptors? So I'm going to take you through this. You don't need to memorize these results. It's more of a big picture thing, but I'm also going to introduce a few things to you. So here is a cell that he is recording from. And the cell that he is recording from, the black sort of trace that you see at the top, which is what I'm pointing to right now, is when he looks at a control tissue, so the cell that does not have a gold bullet embedded within it. So the other thing about biolistics, you can actually look under a microscope. You can actually see which neuron has a nucleus that has a gold bullet staying within it, because that gold bullet doesn't disappear, it stays in the tissue. So you know that that cell is going to be transfected or have the DNA. The one beside it that doesn't have a gold bullet that you can see under a microscope will not be affected. So when he looks at a cell that is not infected or not transfected, he will see this really nice response. And he sees this in the presence of a drug which is known as NBQX. So he's applying glutamate. He sees this really nice electrophysiological response on the left, and he's doing this in the presence of a drug which is known as NBQX. And NBQX is where we have a specific antagonist to AMPA receptors. Because remember, we have two things that can bind to glutamate, AMPA and NMDA. In this experiment, all he is doing is he is targeting the deletion of GLUN1, the NMDA receptor subunit. So in the presence of NBQX that blocks AMPA, he still sees this really nice response that you see over here. When he then goes in on that same cell, and now he applies AP5, this is the response that he sees, nothing. So this is under normal conditions. So AP5 is an antagonist for the NMDA receptor, and so he's able to show that his slices have both glutamate receptors, both AMPA and NMDA. So when he goes back in And now he has used CRISPR-Cas9 to delete gluN1, the prerequisite subunit for the NMDA receptor. He should see an effect on the NMDA receptor. So one, on a Western blot, he definitely sees gluN1 is gone. Like on these two different lanes, the gluN1 unit is gone. All of the other subunits that he hasn't targeted are still there. And then when he goes in and takes a look at the effect of CRISPR-Cas9, in terms of getting a size of current that he would expect, he actually sees that the targeted deletion of the gluN1 subunit leads to the complete loss of the NMDA receptor responsiveness. Not unexpected. You take out one of the key subunits, it just doesn't form the receptor, and that's why he sees like no response. He then takes some of those cells, and this is important, and I want you to make sure that you understand this. When he goes in and he examines the different cells that he has infected, and he does sequencing on those cells, so this is sort of the PAM sequence, this is sort of the targeted region that he wants. When he goes back in and examines why they're not responding, has he actually affected the gluN1 subunit, he's seeing two different things. Or on this one, he's mainly seeing one specific effect, and all of the ones up here at the top. After using CRISPR-Cas9, you are creating a double strand break. You are always going to get it repairing itself again, and that's the non-homologous end joining. And what he is showing is that the reason why the gluN1 subunit is being disrupted, after CRISPR-Cas9 delivery, with the guide RNA specific to this region, he is seeing a deletion. There is a deletion in some of the expected nucleotides. So he sees a deletion here, he sees another deletion here, he sees a much bigger deletion over here. The NMDA receptor subunit is not just targeted, but the genome is actually being affected. He also sees very rarely an out-of-frame insertion. So it makes it out of frame because he's added an extra nucleotide. Both are ones that we would expect to see. After any type of CRISPR-Cas9, every paper that you analyze, they should always go back and be verifying whether there's an insertion or a deletion. We call those an indel. And we see that in his experiments, we always see these indels in the cells that he sees a lack of responsiveness. So again, not surprisingly, and you don't have to worry about all of the details on this slide, I'm just going to take you through one result specifically. The most important one was he wanted to target NMDA receptors. He just did that successfully on this slide here. And he also makes use of some of the pharmacology that is on this slide. This pharmacology is one that I expect you to know. These drugs like AP5 versus NPQX are ones that I expect you to know. The other targeted subunit was the AMPA receptor. The AMPA receptor is normally 4 subunits. Do you remember what one subunit is always required or is found in the vast majority? 99.99% of all AMPA receptors contain 2 of which subunit? The GluA2. And if you have GluA2, what does that mean? The AMPA is not going to allow it to enter into the cell.
Calcium.
So no calcium. So his idea was, let's take out GluA2. What happens to the AMPA receptors? Does it simply disappear, fall apart, vanish, completely go away? Or do we get it to do something else? And again, just to make the long story short, one of the things that he saw, and again, you don't have to worry about any of the results here. If you're interested in them, I'll be happy to take you through them. Using electrophysiology, one of the things that he was able to see is that after taking out the GluA2 subunit, which is what you are seeing here in panel C, he actually saw a change in the currents, or the change in the ions that were flowing through the the AMPA receptor. And one of the things that he saw was that if you eliminate the GluA2 subunit, one of the natural consequences of this is not that the AMPA receptor disappears, is that it gets replaced by other subunits or other genes take over, and now the AMPA receptor could actually cause calcium to flow through using electrophysiology. And again, I don't need you to spend too much time on it, but the results are not unexpected. Taking out a gene that causes a receptor subunit that normally blocks calcium from entering, and removing that now allows calcium to enter. So both experiments were pretty simple, to be quite honest. They were published in a really high impact journal, but they were published mainly because it was the first paper to come out in neuroscience. So these are just proof of principle experiments. They're nothing spectacular. That these postsynaptic density proteins, even though there's so many of them, can actually be targeted using CRISPR-Cas9. The results also, even back then in 2014, they were long-lasting. So even if you were to not do these experiments right away and you were to wait like a couple of weeks, once you knock down a gene, it stays knocked out. So again, a slightly different variation of what Eric Olson saw. And even back in 2014, there were very few off-target effects. He only saw changes in gluN1 when he was targeting gluN1, nothing else. And when he was targeting AMPA, he only saw AMPA receptor-mediated effects. Nothing else in the genome seemed to be affected. So I'm going to just, again, switch gears really quickly, and then the last 5 minutes of today's lecture is just to take you through Fang Zhang's paper, just as an overview, because we're not going to spend a lot of time on this. Fang Zhang's paper, in contrast to Roger Nichols' paper, again, just like 1 year, January 2015, is so different, and it is so much more detailed, and it has so many important implications. And it's done in a living mammalian brain, which is ultimately the hope that we all have, is a spectacular paper. In fact, it should be published in something, not to downplay Nature Biotechnology, but it should have probably been published in Nature or in Science or some other higher impact journal. So I'm going to just mention last year, I'd asked as sort of a bonus question what SP-Cas9 was. Some of you have been asking about this. When we look at the Cas9 nuclease, it is derived from a type of bacteria which is known as Streptococcus pyogenes. That's where it was first cloned from. That's what we still use for most of the experiments that are done today. These are the types of things that, when delivered to eukaryotic cells, cause these insertion-deletion mutations that we've just been talking about. And by putting things out of frame, that gene is not going to be expressed, and therefore the protein is not going to be expressed. This is a large gene. We know that this Cas9 nuclease from Streptococcus pyogenes is fairly large. And what people have been trying to do is to make it smaller and smaller so that you can package other things like guide RNAs into the same vector. Because this takes up most of the space in a vector, it made the original experiments quite challenging. So again, one other aspect to neurons. Neurons are actually postmitotic. When you hear that word, what does it imply automatically to you? Yes? So they're non-dividing. So their DNA is pretty static. That's it for them. And so as a result, their ability to modify their DNA makes it more challenging. And outside of the Nickel paper, there weren't really a lot that kind of looked at this particular technology. So I think I'm going to skip the last slide, which is next, but I'm going to take you through this one slide. And there are some elements that I need you to know on this slide. Again, I'll point out what you need to know. So this is from Fang Zhang's paper. And Fang Zhang's paper is also using 2 different vectors to deliver both the nuclease as well as the guide RNAs. And this is the guide RNA. So 2 separate viral vectors. Nowadays, we package them into one. Part of this is if you take a look at the sequence that we have here in the adenovirus, the vast majority of this is taken up by the nuclease itself. It is a large gene. We can't package much more into this particular virus because the gene for the nuclease itself is incredibly large. So this Streptococcus pyogenes, or SP9 Cas9, is very, very large. We now moved away to the Staph aureus version of Cas9, which is much shorter. It's about half the size, so we can package other things into it. But this is why we have this dual vector system that is implicated here. The only other thing that I need you to be aware of, and this will be the last thing that I talk about, the last slide, the next one, don't worry about it, you won't need to know it for the term test, is that we need to make sure that it is being specifically targeted to the tissue that we want. In the case of Eric Olson, he was using general promoters. That's not a really great technique. It was great because the virus was myotactic, or it was myotropic, it went to muscle tissue, but generally speaking, we want to ensure that if we are targeting a nuclease to a tissue, that it's only going to be expressed in that tissue. And one of these promoters that is specific to neurons is this MECP2 promoter, which is why we have it. It's specific to neurons, and this MECP2 gene and the proteins are usually expressed in neurons. Synapsin, or SYN, which is what you're seeing here, is also very specific to neurons. And so these promoter regions are ones that we now spend a lot of time engineering. So let me ask you this question, and you need to think about it, and it's an application question. You actually should know the answer if you have been studying. Instead of using a myotropic virus that was Eric Olson's design, so he used AAV9, he put it all over the place, it targeted muscle tissue. What specific promoter would you use to ensure delivery and expression within muscle cells? You have a few options. Any ideas? Yes.
Creatine kinase?
So we could use the creatine kinase. Do you remember that from lecture 1? And that's specific, and it's released from damaged tissue. And again, if we're targeting muscle tissue, think about it, because it will be something you'll see. What sort of promoter system could we use to ensure delivery of a nuclease to a specific tissue type like muscle? So think about that for Monday. It's a big sort of indicator for you. This slide you do not need to know. If we have time, we'll come back to it, but we're going to switch gears after term test 1. So we have a couple of options. We can take a really quick 5-minute break, or we can just plow through. Do you have a preference? Plow through? It'll take about 15 minutes. Apologies on that, but we'll just go through. This slide you don't need to know from term test 1, although a really cool slide, and you can see that every single paper you should be seeing this diagram where you're always looking for insertions or deletions. If CRISPR-Cas9 has worked, there always has to be an insertion or a deletion. And it should be near the region that you are making that guide RNA to, and you're seeing that in Fang Zhang's case, he does definitely show insertions, deletions. One of the things that you need to indicate for the upcoming term test as well. So some of you are already spending a lot of time here because I'm actually tracking how you are logging into Quarkus, and this is not what you're thinking. This is Procrastination Island. So if you're thinking that you can spend the weekend, because some of you are, studying for term test 1, I'm going to be honest with you, you can. You can probably still do really well just by studying on the weekend for term test 1. The problem is all your other courses are going to suffer because that's all you're going to be studying. It's probably not a good idea. Take at least some time this week to get 1 or 2 of the lectures out of the way. Come back and review lecture 3 on the weekend. Don't leave all 3 to the weekend. All your other courses are going to suffer. Right beside Procrastination Island is Disappointment Island, and that's somewhere we don't want any of you to go. So you're going to find that, and I'll post all of these slides for you so you don't have to worry. It'll be there. It's also being recorded, I'm assuming. Yes? Okay, and we'll post the recording for you, both the audio as well as the full screen. I'm going to be honest with you because the exam is a little bit longer than last year's. So it's the same content, same sorts of things. The emphasis is a little different this year than it was last year. So for that 60 minutes, I kind of wrote it so that you would spend about 40 minutes writing it, making sure that you have most of your answers. You won't feel too rushed. You're going to probably spend about 10 minutes kind of reviewing it. If you're not good on multiple choice, where it's like fairly long application based, you can look at last year's test to kind of gauge the amount of time that you're spending reading and thinking about your answer choices. It is kind of long. I have designed this to be very fair, but it's also detail oriented on a couple of the multiple choice test questions, as I've already indicated. I've mapped it, by the way, to all of the study guides, and thank you to everyone that's been involved in these study guides, especially the diagrams that are posted for you. They've tailored a lot of the questions specifically around the study guides this year rather than just on what I wanted to do. So I'm going to ask you to stay in EX 320. You can take a bathroom break, but I'm going to ask you to stay there after finishing and in groups of 4 to 5. This will be part of your participation grade. Again, just to put things in perspective, I did go back and check last year's group mark. The average on the group mark out of 5 was 4.7 something. So everyone did really well. I round up, so 4.7 something really means that you got a 5 out of 5. I don't think you can do better than that, but maybe I'm wrong. We'll see, I guess, on Monday. In other words, you can't do better than 5.5 out of 5. So hopefully that's something that you're all aware of as well. If a slide has bullet points, please study them. If a slide has figures, make sure that you know what points were covered on that. Not everything on the slide is going to be covered. So think about it this way. You can break it down in terms of your studying in 2 different ways. One is by topic. We only have 3 topics. Duchenne's muscular dystrophy, dystrophin protein itself and its genes and its complexes, and then genomic editing techniques that we've just been covering. The multiple choice and one-line answers are very application-based this year, a little bit more so than last year. Because we had a pretty good turnout up until today, I will actually post on the announcements page, and this is why I'm telling you not to procrastinate. Some of you log on once per week, probably not a good idea. I'm going to post on the announcements page one of your questions in advance that's application-based. It'll be the identical question, and you'll be able to see it, and I want you to know like how application-based it is. You can discuss the question online, that's up to you, but it's a good idea to make sure that you go over figures presented in class and in the study guides. So I'm using this as an example. I didn't cover everything on this diagram, and I'm not going to ask you questions on things that I didn't cover. So we talked about, for example, this dystrophin protein and how it interacts with actin, and we talked about how some of these different rod domains overlap or are replicated, duplicated, etc. And I also talked about the fact that this will connect to the extracellular matrix, which we didn't go into a lot of detail, through this dystroglycan complex. But I did talk about some other specific things as well, and I mentioned them in passing. So please make sure that you understand that you don't have to memorize things that we didn't talk about in class, so don't panic about that. We spent most of our time looking at this, and this is the structure that we've been looking at throughout this course. Some of you were asking about this diagram, so I thought it was actually a really good time to go back and review this diagram. This is that complex that I showed you earlier. This is the normal muscle tissue, and this is the normal muscle membrane, which we call the sarcolemma, and this is the muscle membrane that you are seeing here. For the most part, when we are looking at a normal dystrophin gene and normal dystrophin protein expression, the calcium homeostasis is really well maintained, and I've mentioned that in that particular lecture. One of the things that I mentioned again in passing, one of the molecules that is associated with this is this NOS or nitric oxide synthase, and this is again mentioned here as well. And I didn't go into this in a lot of detail, but I wanted you to be aware of the fact that the loss of this complex and the loss of this NOS often leads to an overproduction or an increase in the intracellular calcium. So one of the things that you will see, and you have to think about this, I never mentioned it in lecture, if dystrophin isn't there, are the levels of calcium expected to be higher intracellularly or not? So again, you can look at this diagram, and this would sort of be one of the types of applications that I want you to think about. Based on this, and even from the bullet points that I covered in lecture, you would expect there to be much more intracellular calcium in a muscle cell that doesn't have dystrophin, right? So a lot of that is mediated through the loss of those regulatory elements, including things like nNOS, which is no longer there. Beyond that, you don't need to worry about structures like the sarcoplasmic reticulum or any of the other things in this diagram. Again, just to make sure that you are aware. You are not going to be asked to draw the structure of what dystrophin looks like. I don't really care if you've memorized that it has 79 exons or not. If you did, great. It'll be a great trivia question someday, somewhere, I'm sure. But you're not going to be asked for specific numbers like that. It helps because you might get like the MDX mouse model. You can include like exon 23 in your answer if you want, but I'm not looking for specific details. I'd rather you know about the technique and how to analyze it. You should know, though, what utrophin is, and you should know like why utrophin can actually replace dystrophin when it is not present, but I do not expect you to know the structure of utrophin either. Just sort of as a revertant form of this. So some of the other things that you should probably make sure that you know, especially for the multiple choice, go over all of the different animal model systems. We've talked about a number. Zebrafish, for example. We've talked about mice models, dog models, or canine models. Today we talked about hippocampal slice models. Make sure that you know how to identify specific cells or different processes that are going inside specific cells. So for example, could I use H&E staining to identify necrotic tissue? Yes or no? Yep. So hopefully you will know those types of processes using different stains as well as markers. Again, think about the different techniques and their similarities as well as differences, especially for a short answer question. There are a lot more of them than you think. We covered RNAi and we covered morpholino oligos, and we also covered Duchenne's muscular dystrophy. Is there anything that you can think of off the top of your head right now that links all of these things together? Anything? Any, like, just out of the blue guesses? So one of the things that you might be thinking of, especially when we talk about these morpholino oligos. Do you remember there are two mechanisms that were morpholino oligos knock down gene expression by?
Translation blocking and splice blocking.
So we talked about translational 5' UTR binding as well as spliceosome site binding. And today we talked a little bit about spliceosomes and different splice enhancer sites. So a lot of these things actually do kind of overlap. So think about those, especially for the short answer question. And then maybe this is sort of the ultimate on the spot, maybe really high stress, but hopefully not. Can we use RNAi to actually target Duchenne's muscular dystrophy? And maybe that's something to think about in advance, or using these different techniques, and give a rationalization, either yes or no. And sometimes it's uncomfortable kind of knowing those, that you need to be able to justify your answer, but that might be one of the short answer questions that you need to think about. And I just did a hard blank for those of you who are kind of watching, so hard blanks mean something. So think about that in advance. So also, I don't need you to worry about drawing out the structure of a morpholino oligo, but I will tell you that it is a really good idea to study this diagram, know which side of this nuclease has the Cas9, which side of this involves the PAM sequences, which sides of these involve binding the guide RNA, which nuclease will be attacking which strand. It's a good way to make sure that you're not feeling anxious. I'm going to tell you in advance that this is one of your multiple choice test questions, very application based. I've created a guide RNA to this specific strand. Tell me like what's going on. If you know this diagram, you won't have any problems with it, but if you haven't studied it in detail, that's one of the questions that you're going to be stuck on. Please don't leave this to the weekend. Like I'm really serious about that. You need to, I know that you don't like October 1st term tests, or maybe you do you? October 1st, not so much. But I've kind of designed this test to give you a little bit of confidence going on to your other term tests. So I want you to have a really good one. you can do like really well on this term test. It's important to me that you do well in all of your courses, not just this one. And so it's not that it's easy, but it is a straightforward test as long as you study. There are only 3 lectures. You can break it up into studying like 2 this week and then 1 on the weekend. But that's up to you. But it's designed to give you some confidence going into midterm season. Please go over the study guides. Your colleagues have spent time on them. Make sure that you know the details from lectures. Check work as regularly. As I mentioned to you earlier, I will post in advance one of the multiple choice test questions, and maybe if you're checking frequently, so if I see that there's good sort of class log-ons, maybe I'll post something else for you toward the end of the week too, but that's up to your class. One other thing, because this is sort of at the beginning of the week, and normally I would have office hours a few days beforehand, are you guys okay doing sort of an online Google Doc study group? So I'll be posting the link for that, and when I'll be available online, that you can ask questions online at a specific time. It'll probably be on a Sunday evening, maybe around 8 or 9 o'clock. Hopefully you're done all your studying by then. Fingers crossed, yes? Done? Because you'll get a good night's sleep, not be late to the exam center, show up on time, not be too anxious. So it'll be sort of an 8 to 9 o'clock online office hour for any of you to ask any questions. You're all free to log on. And outside of that, do you have any other suggestions, questions? Scared? Anxious? Worried? Nervous? Confident? Confident? Yes?